Welcome to this podcast from Neurogastroenterology Motility. It publishes original research and topical reviews on basic and clinical aspects of gastrointestinal sensation and motility, as well as brain-gut interaction.
So welcome everyone to this month's podcast from Neurogastroenterology Motility. My name is Adam Farmer. I'm a gastroenterologist at the Wingate Institute in London. This month it's my great pleasure to welcome Professor David Grundy. David is a Professor of Biomedical Science at the University of Sheffield and has produced some of the field's most seminal research regarding the neural control of gut function. So David, many thanks for joining us on the podcast this month, and congratulations to you and your co-authors on your paper entitled Interplay between mast cells, enterochromaffin cells and sensory signalling in the aging human bowel. So what have previous animal studies shown with regards to visceral pain perception and how this changes with age?
Okay, well, thanks, Adam, for inviting me to discuss our paper with you. We've actually learnt a lot about the mechanisms that underlie sensory signaling from the gut, both in terms of morphological features, association of afferent terminals with other structures in the gut wall, and the molecular mechanisms that determine excitability of signal transduction in the peripheral endings. And in this respect, what has been described is a diverse population of endings, each with their own characteristics. And in these animal studies, it's been shown how they transduce different stimuli, both from the lumen of the gut, but also from within the wall, the gut wall itself. And in the last 5 years or so, these same receptor endings have been seen to be present in human bowel. And in this respect, there seems to be a good correlation between human and these animal mouse studies. So whilst the basic principles of signaling seem to be comparable in our animal studies, little is known from human work how these mechanisms alter with age. Although there is evidence from clinical studies that there is a blunting of sensitivity in the elderly, and this potentially could lead to some of the symptoms of bowel dysfunction that occur increasingly as we age.
So in your experience, what do you think the main determinants of visceral sensation are?
Well, it's a combination of peripheral and central mechanisms. The peripheral mechanisms are there to detect events within the bowel, and in terms of sensation, things like distension or powerful contraction can lead to sensory experience. But you really need the central mechanisms in order to interpret those peripheral signals. And those central mechanisms are then necessary for the perception of the stimulus itself, but also the emotional behavioral consequences of visceral pain. And we know a lot about those central mechanisms in humans from studies involving functional brain imaging. In humans, we know much less about what's happening at the peripheral level in terms of signal transduction.
So what do you think the main weaknesses, particularly the animal studies, are to date really with respect to direct human translation?
There's always going to be a question mark hanging over animal studies in terms of the extent to which these findings translate to humans. And in particular, there have been some concerns recently from drug discovery programs and novel therapeutics that have been validated in animal studies but have then subsequently failed in the clinic, in clinical trials, either because of unpredictable adverse events or for lack of efficacy. And whilst there are many factors that can contribute to failure in translation, species difference is always going to be a concern. And so by being able to work directly in human tissue can remove one of those obstacles.
So what were the objectives of your study?
Well, we've been recording from visceral afferents in isolated gut segments for many years. And about 5 years ago, we started to explore the possibility of doing this in human resected segments. And the rationale was that, if we can record from these nerves in mice, then it should be possible to do in humans. But in humans, there's huge logistic difficulties that limit first of all, the access to the tissue, and also the ability to get viable tissue back into the lab, where it can be studied using our electrophysiological techniques. And it really takes a dedicated team, both the clinical scientists who are involved in the surgical procedures and going that extra step in order to provide us with the tissue. And then in this case, Yang Yu and Wen Yang, who put in a huge amount of effort in order to be able to develop the technique to record from these nerves that's allowed us to characterize features of human gut sensory signaling. The other aspect of human work, though, is that it provides a diversity of tissue. We tend to take anything that's available, and that means we're looking at different regions of gut, segments from males and females with different underlying pathology, different age ranges. And that has allowed us to probe the factors that influence afferent sensitivity in these patient samples. And in this particular study, we've examined the impact of age. And for statistical comparison, we made the cutoff at 65, which keeps me in the younger category just about.
That's brilliant. No, I agree. These studies take a Herculean effort from a number of members of the team. So what were your methods and really your key results from your study?
The main approach was electrophysiology. So the recording of action potentials in the colonic afferent nerves as they emanate from the bowel wall. And we've combined this electrophysiology with the morphological characterization, particularly looking at nerve density using antibodies to label nerve terminals in the gut wall. We've looked at mast cell distribution and also EC cells, and tried to correlate then changes in these different cell populations and afferent innervation. And we've also used PCR to examine expression of key sensory receptors and how they may be different in younger and older samples. And then what were your key results to come from this study? Well, by recording directly from the afferent themselves, the impulse traffic from these samples, we've demonstrated that comparing younger and older specimens, that there's an attenuation in nerve activity with age. And this is associated with concurrent changes in nerve density, in enterochromaffin cell numbers, and also in mucosal mast cells. And what were the limitations of your study? There are technical limitations to this kind of work. Despite the best efforts of our clinical colleagues, the number of viable preparations that we could get into the lab and actually record afferent activity, it was about 1 in 4. And so it really is a slow process in developing the data from these kind of experiments. I guess the main limitation is that, unlike in animal studies where you have defined genetic backgrounds, you have control breeding, you have controls over the food intake and time of day and all of these various factors that help to eliminate some of the variability. We don't have that in these human samples, and so things like genetic factors, medical history, and in particular, I think prior treatment can confound some of the data interpretation. So whilst we see changes with age, whether it's age per se that is defining those changes, or whether it's a consequence of a lifetime of other treatments, and history is not clear.
So where do you think the knowledge gaps are in the field, and how, in your opinion, should we go about addressing these in the future?
I think there's still a great unmet clinical need for new drugs to treat GI symptoms such as pain. Using this type of approach, recording from human sensory afferents directly, means that we can introduce human studies into the drug discovery program at a much earlier time point. And this will help, I think, eliminate some of the question marks around species variability. But it also means that we can move the drug discovery program forward considerably. And in respect to the influence of aging on sensory signaling, then perhaps the findings that we've shown here with the blunted signaling from bowel of elderly patients, this might lead to approaches to help manage GI symptoms in these patients.
So with that, David, I'd like to thank you and your co-authors for a really excellent paper and assisting in this month's podcast, and also our listeners for tuning in. And I look forward to welcoming you all again on another instalment of the podcast next month.
Further information about this paper can be found on the journal website. We hope that you have enjoyed this podcast, and we look forward to welcoming you to next month's edition.